Hello, my name is Dr. Chaim Leibovitz, and on behalf of the GEM project, I would like to provide you with a brief summary of our recently published gut paper titled Immune Response and Barrier Dysfunction-Related Proteomic Signatures in Preclinical Phase of Crohn's Disease Highlight Earliest Events of Pathogenesis. The Crohn's and Colitis Genetic, Environment, and Microbial GEM project is a unique multi-center international prospective study that investigates the pre-disease phase by following more than 5,000 healthy first-degree relatives until some of them develop Crohn's disease. This cohort is deeply phenotyped, and each participant provides baseline serum, urine, and stool samples, which we can then analyze for different biomarkers that can help us distinguish those who develop Crohn's disease from those who remain healthy, and by that learn about the factors that may trigger the development of Crohn's disease, with the ultimate goal of finding ways to modify those factors to try and prevent the development of Crohn's disease. And here is an illustration showing the preclinical disease stage before patients develop symptoms and are already diagnosed with Crohn's disease. And as you can see, we already found some important factors such as the gut microbiome, the gut barrier function, and antibodies against microbial antigens that are already altered years before diagnosis of Crohn's disease. And here in this study, we hypothesized that there might also be a serum proteomic signature associated with future development of Crohn's disease. And additionally, we aim to see if these Crohn's disease risk proteins are associated with the other risk biomarkers, and by that to gain insight on the different pathways involved in the development of Crohn's disease. So what is the serum proteome? So the serum proteome represents the entire set of proteins produced by an organism, and it provides information of expressed proteins involved in physiological and pathological processes of all body tissues, including the gut, and therefore has the potential to yield valuable insight into the understanding of gut homeostasis and to serve as a clinically relevant biomarker. So for this study, we used a subset of the GEM cohort in which we included 71 healthy first-degree relatives who developed Crohn's disease during the follow-up period and matched them with 284 first-degree relatives who remain healthy. As you can see here on this plot, we identified 25 serum proteins that were associated with future development of Crohn's disease, 15 proteins seen here on the right side of the plot colored in blue, including CXCL9, oncostatin M, and several metalloproteinases were associated with increased risk for Crohn's disease, whereas 10 proteins here on the left side of this plot colored in orange, including the anti-inflammatory hemoxygenase 1 enzyme, were protective of future Crohn's disease development. Then, after identifying these set of Crohn's disease risk proteins, we asked ourselves whether they may represent different pathways involved in Crohn's disease pathogenesis. So the first thing we looked at was gut subclinical inflammation measured by fecal calprotectin. And here we can see on the left side that majority of the CD risk proteins were also associated with fecal calprotectin, which we know is a part of the pre-disease continuum involved in gut inflammation leading to Crohn's disease. And as you can see on the right side, you can see that CXCL9 had the strongest association with abnormal fecal calprotectin, whether hemoxygenase 1 had the strongest inverse association with fecal calprotectin. When we next looked at associations with gut barrier function measured by the urinary excretion of the sugars lactulose and mannitol, we were able to identify some of the Crohn's disease risk proteins to be also associated with gut permeability, and interestingly, among which was MMP12, which belongs to a family of proteins named matrix metalloproteinases, which are proteases involved in tissue injury and remodeling, which are important in maintaining the structure and function of the intestinal barrier. And finally, as you can see here in the figure, we found that a portion of the proteins that were associated with increased risk for Crohn's disease were also associated with antimicrobial immune response. And interestingly, CXCL9 was the only protein that was associated with future Crohn's disease risks, and all the other biomarkers of subclinical gut inflammation, gut barrier function, and antimicrobial antibodies, suggesting that this protein may have a pivotal role in Crohn's disease pathogenesis. So in summary, the key takeaways from this study are that the serum proteome are not only predictive biomarkers, but may also shed light on alterations of the underlying biological pathways leading to Crohn's disease onset in the preclinical stage. And these findings provide insight on the possible interactions of preclinical events in the evolution of Crohn's disease development, and may also be potential biological targets for preventive interventions against disease development. We would like to thank all our collaborators, the participating sites, and the funding agencies supporting the GEM project. And of course, to all the participants who took part in this study. And we encourage you to read the full paper for more details. Thank you.